Hi, I'm Nia, and I'm this semester's Down-Combe Research Scholar Co-op out of the Ocean Genome Legacy Center. Today, I'll be presenting on preservative methods for long-term DNA preservation. So, first, some quick background. OGL is a biorepository, which means we have a very large collection of DNA and tissue samples. Some of the ways that we get that is by working with a multitude of different scientists. When they send us their tissue samples, they need to be preserved before they can reach our lab for DNA extraction. This is because DNA degrades really quickly in tissue samples without treatment. So that brings us to the question of how can we maintain DNA quality in tissue samples between collection and processing? So some common preservatives include ethanol, which, like I said, super common amongst many different labs, and even in some museum collections. Salt has been known to have been added to different preservatives to increase preservation. And freezing is a gold standard for preservation. However, it's much harder to do when you're in the field. EDTA is another chemical preservative that OGL has been researching over the past couple of years. So in this project, we're looking to improve the quality of DNA and tissue in samples that have been stored for longer periods of time. So what exactly are we exploring in this experiment? Here, we are comparing 8 different preservation methods and their effects on DNA and tissue quality. So first, we have our fresh and liquid nitrogen treatments, which are our controls. And then we have our single preservative solutions, which are EDTA and ethanol. Then we have our freezer treatment, which is liquid nitrogen flash frozen and then stored at negative 80C. And then we have our combined preservative solutions, which is EDTA and salt, EDTA and ethanol, and EDTA salt and ethanol. In addition to comparing these preservatives, we're looking at where is the DNA found when we preserve these tissue specimens. Is it going to be staying in the tissue itself in that little orange box there, or is it going to be seeping into the liquid preservative in that more light blue area? So to quickly walk through some experimental design, we have 5 taxa that we're looking at. We have the lobster, the sea nettle, clamworm, bloodworm, and blue mussel. After each of these animals have been dissected, their tissue samples were put into one of our 8 treatments. Like I said earlier, our fresh and liquid nitrogen treatments were our controls, and they were immediately extracted from right after dissection. All other tissue samples were left in their preservative types until they reached our time point check-ins. We had 3 time points of 6 weeks, 6 months, and 1 year. Once they reached these times, we did DNA extractions on them as well for both tissue and liquid preservatives, which ended up being quite a few extractions. So thank you for everyone at the MSC who came to our Skittles extraction party. And then once we have the DNA from these samples, we can do some analysis on that. So we looked at gel electrophoresis, tape station analysis, and tissues quality. So before I get into any of the results, I want to quickly go through what we're really looking for. So on our gels, we're looking for high molecular weight DNA bands. So in our good bands photo, we see that top of our ladder at the 10 kb mark, we see very nice, tight, clean lines in our sample wells, which indicate high molecular weight bands. However, when we look at our poor bands photo, we see the smearing down the lanes, which is indicative of degrading DNA. In addition, when we talk about tissue workability, we're looking at how easy is it to subsample the tissue? Is the tissue still intact? And does it smell? So in that photo there, we see this fun goo that's connecting our tissue sample to our preservative, which we lovingly call worm soup. But unfortunately, it's not the easiest type of sample to work with. So our focus taxa for today is going to be Glycera capitata because this is one of the 3 specimens that have reached the 1-year time point we have a full set of data on. So first, we're going to look at some gels for the tissue extractions. First, we have our controls. We see that we can get a decent amount of high molecular weight DNA out of these samples with some occasional smearing. And then we look at our single preservative solutions. We see that EDTA is a bit spotty. We don't see a lot of DNA there, some high molecular weight and some with nothing at all. But when we look at ethanol and liquid nitrogen stored at negative 80, we see that there's a lot more DNA there, but it's a bit more smeary and it's not as clean. Moving to our combined preservative solutions, we see that EDTA and salt is a bit better. It's a bit tighter still, but still a decent amount of smearing and a little spotty there. But looking at our EDTA and ethanol, as well as EDTA salt and ethanol, we see that there are much cleaner, nicer bands around the top of our ladder at that 10 kb mark, indicating high molecular weight DNA. So now that we've looked at a more qualitative analysis of our DNA, we're going to move to a more quantitative analysis. So first, we're going to look at our percent high molecular weight DNA, which is of all the DNA that we extracted from, how much of that is high molecular weight. So with our treatments along the bottom, we see that treatment does not have an effect on the percent of high molecular weight DNA of the DNA that we extract from. However, when we look at high molecular weight DNA yield, which is the ratio of high molecular weight to the overall size of the sample we have, we see that EDTA and ethanol, as well as ethanol by itself, have a significantly higher yield than EDTA by itself. Moving to preservative, we see that ethanol-based solutions seem to have minimal, if any, DNA coming out of the preservative, whereas when we look at EDTA and EDTA in salt, we see much more DNA coming up on our gels, meaning that the DNA is seeping out of the tissue and into the preservative. So what might this mean for what the preservative is doing to the tissue samples themselves? So we're going to look at some tissue degradation and sample workability. So first, we're going to look at tissue texture. So we have our blue is soft and orange is firm. Again, with our treatments along the bottom, we see that ethanol-based solutions are much firmer, whereas our EDTA and EDTA in salt had much softer tissue samples, which made it much harder to work with. Moving from there, we're looking at tissue handleability. Again, with our treatments along the bottom, in orange, our ethanol-based solutions are super workable, very easy to handle, whereas our EDTA and EDTA in salt samples are very difficult. They had that warm soup consistency, which made it very hard for us to subsample the tissue. And lastly, we're going to look at sample smell. Again, with our treatments along the bottom in orange, and we see that there is a minimal smell coming from samples in ethanol-based solutions. Whereas when we look at EDTA and EDTA in salt solution, we start to get some of that stronger, more unpleasant smelling specimen. So some of our final conclusions, solutions with ethanol had more consistent bands on gels. EDTA and ethanol, as well as ethanol alone, produced higher yields, and it overall made the tissues more workable. EDTA solutions without ethanol added to them had much fainter or less consistent bands on gels. EDTA alone had a significantly lower yield than either EDTA and ethanol or ethanol by itself, and overall were harder to work with and handle. And lastly, for our final treatment, liquid nitrogen stored at -80, these samples performed very similarly to the other treatments when comparing against DNA quality and even tissue workability. However, it required really quick processing to reduce the effects of thawing so the DNA wouldn't degrade in that time. So our next steps would be to finish consolidating and analyzing the data for the other two taxa that have reached the 1-year mark, which are our clamworm and our blue mussel. And then final conclusions for this whole experiment will be made after 1 year of the data for all taxa have been collected and analyzed. So we still have to wait a bit more time for our sea nettle and our lobsters. So thank you to everyone. A special thanks to the OGL team, Rosa, Hannah, and Dan, and to the previous co-ops who worked on this project, Ryan and Molly. Thank you all for tuning in, and feel free to reach out with any questions. Thank you.